So it's a great pleasure, obviously, you can tell, to introduce this sequel, Reproductive Biomedicine in Space. This is the trip to Mars. Investigations into the effects of space have been going on for decades, as more and more people are working in space. Indeed, since the year 2000, humans have been living constantly in space. In the first webinar, which was in October, session 37, if you wish to look at it on our website, we investigated experiments from the International Space Station, highlighting the reproductive hazards of space travel for men and for women in the presence of increased radiation and microgravity. In fact, many of the observations are relevant to us even now on Earth and give us insights to the more terrestrial problems of earthbound infertility. And now, of course, there are plans to go further. In fact, President Biden has recently endorsed the effort to go to the moon. NASA's human exploration plans with the Artemis program call for sending the first woman and the next man to the surface of the moon by 2024. And plans are vigorously underway to go to Mars. So before I hand over to the experts of space medicine, I'm joined by Jacques Cohen. And those unfamiliar in the IVF world, his Twitter feed reads, clinical embryologist, product developer, US citizen, Dutch citizen, US resident, I should say, perhaps a citizen of the world, vegan, atheist, step counter, and trivial collector. Well, he's a reproductive biologist, high complexity laboratory director, and he's director and founder of the Art Institute of Washington. He's co-founder of Reprogenetics and Life Global, IVF Online 2. The same role he now commands with IVF QC Althea Science, which develops software for labs and clinics. He's Emeritus Chief Editor of Reproductive Biomedicine Online, but not in space. He has co-developed a bunch of embryological methodologies and devices, and currently serves 6 scientific advisory boards. So you can look at that on the website. So you name it, he's done it, but not in space yet. According to Intel I have, he even gets sick when he's driving himself. So he'd probably fail the first day of space camp. So over to my science officer. Over to you.
Thank you, Commander Palmer. Yeah, this is as close to space I will ever get. It's a real pleasure to be co-hosting this session with you, and welcome everyone. For those of you who do not know our Commander Palmer, before he went off to space on I3, he was a clinical embryologist. He was, and he's skilled in laboratory, business, and quality management since then. After graduating in genetics, he's a geneticist by training at Leeds University. He worked as a research officer at London Hammersmith Hospital, very famous IVF unit, and he worked together with Lord Winston, Robert Winston, and Professor Alan Handyside. And some of their madness is still what you can notice nowadays in Giles. He has published in leading scientific journals on a variety of topics, including PGT, on cystic fibrosis and beta thalassemia. He's also published in Morphokinetics and Quality Management. He is an HCPC clinical scientist. I'm not sure what it is. I'm sure it's very important. And has been accredited with senior embryology status by ASHRA. I know what that is. He's an executive director of I3. He actually is the only executive director of I3. And he is responsible for everything, okay? He works 24 earthly hours a day, putting all these sessions together every week, and now going to Asia sometimes twice a week. I hope you don't suffer from space sickness, Giles, like I do. It's now my pleasure to introduce to you the first of 2 of our panelists, Dr. Fathi Karouia. Fathi was with us in October in session 37, and his lecture is still on our website. Fathi is the real thing. He's a senior research scientist within the Astrobiology and Space Sciences divisions at NASA Ames Research Center in Silicon Valley. Can you imagine you work for NASA and you live in Silicon Valley? Really, that's just incredible. Dr. Karouia conducts research related to astrobiology, space biology, human spaceflight. In particular, he's interested in life beyond Earth and how biological systems respond to the space environment. He's also interested in understanding and mitigating the effects of space radiation and astronauts' health. and developing high-throughput technology for in-situ space research. Now, I hope you're sitting down. Dr. Fathi Karouia received a PhD in biology from the University of Houston, a master's degree in space studies from the International Space University, a master's degree in space technologies from Louis Pasteur University, a master's degree in physics and an embryology degree, and a bachelor degree in physics and chemistry from Henry Poincaré University. He obviously collects degrees. It must be very, very easy for Fathi to do this. What about getting a degree in xenoarchaeology next, maybe? There's no limits. It's just amazing what an incredible CV you have. We have a question for you. Giles has a question for you. What keeps you awake at night when you consider the physiology, Fathi, of manned space flight to Mars? What is the first thing that comes in your mind that would make you really, really worried? And I know this probably could take you hours to answer this because you have all this information, but what is it particularly that would worry you?
Well, first of all, thanks again for the introduction, and I'm very delighted to be part of this session again, and so thanks for the invitation. In regards to the effects of space environment on humans, I mean, obviously a lot of people and few of the panelists are hardly working on those type of topics. I think for me, most likely radiation might be one of the main limited factor for long duration space flights. And even more when we talk in relation to a mission to Mars. In relation to biomedicine and reproductive medicine, I mean, as you know, I was part of a team which we flew for the first time human and bovine sperm in space. And the idea there was to understand how sperm is behaving under microgravity condition. And so actually the main aspect of the study was trying to assess if a sperm can capacitate under microgravity condition. And as you know, capacitation is a very important process to be able to fertilize the egg. And that requires a series of changes in regards to motility parameters, changes in regards to protein modifications, metabolism, and also morphology. And so actually we realized that the impact of microgravity was significant on the motility. And more importantly, the acrosome rate reactions for human and bovine sperm were also decreased. What is interesting in relation to space radiation, our samples were stored for roughly 4 months on board the International Space Station prior to the operation to occur. And we realized that many of the samples actually were damaged, which means that their viability were impacted. And so it is possible that is a combination of storage, but most likely it was probably the impact of ionizing radiation. So I would argue that radiation might be one of the main factors in regards to long duration spaceflight. And I'm looking forward to hear from Professor Mason in regards to what can be done at the human level to try to mitigate those risks. And so maybe engineer a Superman that would be able to cope with the environment, the lack of gravity, and the increased level of radiations and so forth.
Okay, that's great. And that's a bit of information which you didn't mention in the first session, of course, about the fact that it could be damaged. And of course, we are going further away from low Earth orbit. So we're definitely going to have lots of questions for you. We have, in fact, questions from last time, would you believe, as well, which are going to slip in there as well, people that didn't get the chance. But I'd like to introduce our other guest panelist. It's Professor Virginia Wotring, and she was with us last trip, if you like. She's been using her training in biochemistry, physiology, and pharmacology to study the actions of medications using and during spaceflight missions. This means first studying the effects of spaceflight environment on physiology, and then looking at the changes in pharmacokinetics and pharmacodynamics. She's been doing this in positions at Johnson Space Center and the Bayer College of Medicine in Houston. And now she's professor at the International Space University in Strasbourg, France. She's held management positions in the past, most recently a deputy director and chief scientist at the NASA-funded Translational Research Institute of Space Health. So I'd very much like to welcome Virginia here to the panel. And can you, Virginia, can you tell us a little bit more about the International Space University? And in fact, how can I enroll, to be honest?
Absolutely. Thank you so much for inviting me to join you again. The International Space University is exactly what it says. It's all about space. It's a university devoted just to space questions. Now, it's not just astronomy or physics or engineering that you might think of being associated with space, but we also have in our course equal weighting on what happens to human beings in space, physiology and pharmacology that I teach, and also ethics, government policy, the humanities, and how space affects what we do in art. So it's a very interdisciplinary kind of a program, which I think is really different from how most people think you need to be educated to work in space. We think of space as very, very sciencey, but ISU is making it a well-rounded venture that we could actually move into for the next part of our history.
Okay, thank you very much.
Thank you, Virginia. It's my pleasure now to introduce our first speaker, Dr. Christopher Mason. It's a real honor to introduce you. He's an associate professor of genomics, physiology, and biophysics at Weill Cornell Medicine and the director of the World Quant Initiative for Quantitative Prediction. He also holds affiliate appointments at the TRI program on computational biology and medicine, which is Cornell Memorial Sloan Kettering Cancer Center and Rockefeller University. The Mason Laboratory is amazing. It develops and deploys new biochemical and computational methods in functional genomics to elucidate the genetic basis of human disease and physiology. The incredible studies performed there are detailed in his bio section on our website. As part of his interest, he also works with NASA to build integrated molecular portraits of the genomes, epigenomes, transcriptomes, and metagenomes for astronauts, which help establish the molecular foundations of genetic defenses for enabling long-term human spaceflight. He is a transformative R1 NIH award recipient. He received the NASA Group Achievement Award and the World Quant Foundation Scholar Award. He is the recipient of many other awards, which you again can look up on our website. He was named as one of the brilliant 10 scientists by Popular Science, featured as StatMed speaker, and called the genius of genetics by 92Y. He has authored more than 230 peer-reviewed papers, and he's published and is on covers of Nature, Science, Cell, Nature Biotechnology, Nature Microbiology, and Neuron, as well as legal briefs cited by the US District Court and US Supreme Court. Many presentations in public as well. He's an inventor of patents. He co-founded 5 biotechnology startup companies, and he sits as an advisor on many others. Dr. Mason, pleasure to have you here. And we're looking forward to hearing you talk about the impact of spaceflight on the human body and preparing for Mars missions. If you could just unmute.
All right, great. Thank you for the wonderful introduction. You'd think it was my first time using Zoom, but it is unfortunately not. All right, can you hear me and see slides okay? Looking all right?
Yeah, looks good. Thank you.
Great. Wonderful. Well, thank you so much for the introduction. It's really a pleasure to be here. It's an extraordinary group, and I'm really honored and humbled to be with you all here today. I'm going to present some work that's come from our laboratory in the past few years on understanding what happens to the human body at a very fine-grained molecular scale when you're in flight, and then also how we can use these lessons to prepare for trips to Mars. I'll also put my slides up online on Twitter. This is my Twitter handle. Twitter is an important platform for sharing science. It also used to be a major platform for United States policy announcements by our former president, who's no longer on the platform, however. So now it's just a fun platform for sharing science and ideas, I'd say. Okay, so I will point out that really there's this extraordinary new rush to get to the moon and to Mars with NASA to thinking about getting by 2024, as you just heard, to have the first woman and the next man walk around the surface. Might be 2025 based on recent changes, but it's coming soon. And this is also part of a long-term goal. You know, the moon is just the first stepping stone to get towards Mars, where we could deploy many technologies and scientific approaches to really enable new exploration on the red planet. And it's worth noting, actually, in 2 days, we'll see actually the Perseverance rover will be, if all goes well, landing right on the surface. And so, you know, this is a lot of excitement, of course, from within the United States, but it's not just the US that wants to go there. China and India are racing there as well. And you can see here, there's really kind of a new space race. And in some ways, it's like the 1960s and 70s all over again. Including you know, players that have not previously been in space, including, for example, the Israeli spacecraft that landed on the moon. In this case, it actually crash landed though, and actually put water bears or tardigrades on the surface of the moon in August of 2019. These are these kind of cute little water bears that can even survive in the vacuum of space, and I'll come back to them later. Other organisms that have recently landed on the moon include plants. This is from the Chiang 5. Actually, cotton was growing on the moon. And so you cannot yet get your cotton pants from the moon, your lunar pants, but you could imagine someday that might be happening. And then the other thing that's interesting is there's even now ESA has been giving out grants to architectural firms to look at what will the moon bases look like in the near future. And so this is actually a rendering from one firm from Skidmore et al. And you can see this is actually a future that's not that far away. So you can imagine if you look at any children you know today and they say, I want to grow up to be a lunar architect, you can actually say that is a legitimate job hope, because people have that job today. So it's really an exciting time to think about this, not just as a quick visit and planting a flag, but thinking about a long-term settlement there that we could use as a springboard to go to Mars. However, we know that going to space has its risks, has its stresses on the body. That's why, of course, you're here. And we know this, we've known this for a long time. These are actually some of the first recordings of basically these nuclear foil packs that Neil Armstrong wore around his ankles that measured the high energy particles zipping through the foil and therefore zipping through his body. And you can see here the traces of the high energy particles that left in these foil packs. And we know that there's this radiation risk, which you heard about, and there's also, you know, other risks just of being in microgravity or lack of gravity, and also just in general the stress on the body, the distance from home, all the stressors of spaceflight. And so what we want to do is understand, what is the real radiation risk? How do we measure it? How do we even try to think to prevent it? And then also, how do we put this into the context of what else changes just as we age? So for example, we know that telomeres shrink as we age. They just get shorter as a function of time. This is something you've probably heard about and learned. We also know that there is epigenetic drift, or DNA methylation is one way. If you look at identical twins at 3 years old or 50 years old, you can see they have many more differential methylated bands. Their DNA methylation, or their epigenetic states, change and drift apart as they age. We also know that people accumulate mutations just as we age. One aspect of this is called clonal hematopoiesis, is that we all eventually accumulate these mutations in our bone marrow, and eventually this can even lead to risk of leukemia or cardiovascular disease, something called clonal hematopoiesis of indeterminate potential, or CHIP. And there's a really example, some beautiful work that was done at Cornell on this. And so we know that these are the things that change just as we age. But, you know, what about in space? This was a big question. We had a unique opportunity because you could see Mark and Scott Kelly, identical twins. One was going into space for a year, and this is all now. Mark Kelly is now also a senator. So we have the beginning of the Senate Genome Project in our laboratory. You can see here. And we had a unique opportunity because Scott was going over for the longest ever NASA mission, a year in space. And we launched a mission with many other collaborators. We were one team out of the 10 teams coordinating the study for this mission. And it was a really exciting time because we could study what happened over the course of a year. And we did get, I recommended epigenetic spots to be on the patch. You can see the little circles in the DNA. I was very excited about that personally. And so after Scott was up in space for a year, you can see him here undocking from the space station. You can see the Soyuz capsule, you know, drifting away in space. You can see the Earth floating down below to the lower right. And if you can read the Russian telemetry data, you can see his trajectory. And eventually, you know, made it into the atmosphere. The chute deployed just fine. So he's actually making his way through, landed in Kazakhstan, was helped out by a lot of Russian cosmonauts as well as other support staff, and kind of eventually got back and was wonderfully gave us kind of a thumbs up when he got back. But when he got back to Houston, he felt like actually his skin really felt like it was on fire. He had rashes and discoloration anywhere he touched anything. he basically had to almost walk around his house naked for a couple days, which, in our family, we just call that Sunday, but he had to do it for medical reasons. I mean, he also had flu-like symptoms. He wanted to go to the emergency room. He really felt awful. And of course, he knew he'd just been in space for a year. But at a molecular level, we wanted to understand why, what had happened inside his body that could help us better understand what happened to him and prepare for future missions. So in particular, we wanted to look at everywhere we could. When you don't know where to look in the body for something that's causing discomfort or disease, you want to look everywhere that you can to try and map it. So we looked at genetic variation, epigenetic changes, RNA expression, RNA methylation, proteomic, chromatin, antibody titers, B cells, T cells, telomere length, cytokines, metabolomics, microbiome, cognition, and vascular metrics every about basically 2 to 3 weeks over the course of about almost 3 years to bring us a multi-omic analysis of human spaceflight. So by multi-omics, what I mean is we looked at all the things we could and looked for things that went up and down together at the microbiome, the genome, the transcriptome, the metabolome, even looked in, so go back to telomeres. We looked at everything we could to see things that went up and down in concert with each other. And one thing you can see here that's kind of striking to begin with is that his telomeres got longer in space here. You can see versus controls or versus his brother, they actually increased in flight, which we thought was really surprising. This is work with Susan Bailey. And we then validated this in our lab as well as in her lab with multiple extractions, multiple sequencing methods, and then confirmed this again in additional 2 other astronauts that we just published last year. But then since then, we thought, well, what's driving this change? And so we thought, is it oxidative stress? Is it the response to maybe amounts of radiation at a subclinical level? In other places where there's stress, and we thought even in some cases hypoxia, is, for example, when people go on the top of Mount Everest. It's the closest you can get to the space station while you're sitting on Earth and standing on the Earth. So we thought, well, let's study, basically astronauts and compare them to mountaineers. And here too, we saw the same thing. Actually, people climbing Mount Everest, we got actually twins, some who stayed at sea level and some who went on the top of Mount Everest. They also showed longer telomeres. So we can see this combined factor of both, essentially physical stress, low dose radiation, it's just getting up to the top of the mountain, seems to lead to longer telomeres in a temporary fashion consistently. And so this means that, Scott Kelly got technically in some ways by genetically a little bit younger, and he also got a little bit taller. But did that matter for his epigenome, his DNA methylation? So when we looked at that, actually, we could see that his age, here's Scott in blue, stayed actually about the same over time, whereas Mark on Earth in green increased a little bit, but also stayed pretty flat. So they're both relatively healthy, and this didn't seem to drive their epigenetic age to get too much older or younger. They actually stayed pretty stable, which is good for their age. That was in some sense kind of, good news. And then we wanted to then also look at what about mutations in his blood, that clonal hematopoiesis question. Could we observe anything there? And here, what was really also surprising is clonal hematopoiesis got a little bit better in space, it actually decreased. This is the allele frequency of the number of sort of cells in his blood that were carrying the mutations. This is for TET2. And before we went in flight, it was at almost getting up to almost 4%, but it dropped down and stayed down afterwards. So I thought this is actually the number of mutations in his blood that looked like they could be, ones you'd want to keep an eye on, got a little bit better. So that was actually kind of good news. But when we kept track of them, we've been tracking them ever since the mission, we could see that it actually did come back and increased in the years afterwards. So we're continuing to take samples every 3 to 6 months to maintain sort of a tracking of both Mark and Scott, as well as expanding this to other astronauts, which I'll show you just at the very end of the talk. And so this just published also in November last year. Overall, so we've seen, these are some of the stresses on the body, but there's other stresses that we've seen, not just in astronauts, but also in animal models. This includes mitochondria that not only are transcriptionally perturbed, but also we see they're literally ejected into the bloodstream or present much more than we see in controls as a consequence of some of the spaceflight stress. So we can see fragments actually in the blood that we can actually infer which cells are dying. Most of them are actually just normal blood cells, which is good to see. But we also found more brain-derived exosomes in the blood of Scott than we've seen in other astronauts or any other controls as well. So we do see, interesting changes that might be potentially long-term that we can observe, and we just published some of these again a few months ago. But I want to jump back to some of the other things that might be driving this, right? So I mentioned a bit of hypoxia, the stress on the body, the radiation, the lack of gravity. All these are factors for sure. And so to better tease out what might be driving some of this, we looked at the gene expression profile. So in particular, you can see Scott here in blue and Mark is here on Earth. You see in uncolored. This is 2 different kinds of RNA prep. So we did these what's called a ribodepletion versus a poly-A prep. So 2 different ways to examine all of the types of RNA that we find in the blood. And we also sorted these cells for CD4, CD8, CD19, and lymphocyte depleted fractions. So multiple cell types examined from the blood that we sorted. Some of these were sorted and separated in flight and then further separated on Earth. But we can see here, overall, we can see these bursts of purple and orange are genes that are going up or down in terms of in flight. And then you can see the color goes away when he gets back on Earth, this green part. So it means that genes go up and genes go down, but for the most part, they return to normal. But about 8.7% of these genes didn't return to normal even 6 months later. So we thought, well, what are those genes? What are these kind of space genes, as Trevor Noah called them at one point? What are these genes? And so we actually looked at the changes in gene expression that are there after the flight include the immune system, DNA repair enzymes, and even some genes for hypercapnia, which we were surprised about because the space station is very well regulated for oxygen and carbon dioxide. But Scott even complained about it a bit in his book, that there was some levels of discomfort from too much carbon dioxide. So then we asked other astronauts and talked about what could be occurring. And there was a discussion that if you don't have enough airflow by your face, you can actually get a bit of small clouds of carbon dioxide that stay by your face. This is actually best exemplified by astronaut Jack Fisher. When you blow bubbles in space, they don't just go away from your face, they actually can stay even near your face. So we think small clouds of carbon dioxide might be leading to some of the sort of blood-derived hypercapnia signatures we see in the blood sequencing. And so it's not only that, but also we see other stresses on the body that might have led to some of these changes. So for example, when he landed back on Earth after a year, some of the things that we've seen is that the immune system rapidly responded and even very much dramatically to returning to gravity. For C-reactive protein, interleukin receptor antagonist 1, sometimes increasing 1,500% and also above the baseline you can see for himself as well as relative to his brother. And so we know that these are some of the signatures that we are now looking at for some ways of pharmacological interventions or things that we should keep an eye on for future missions. So I want to close on one last point about the twin study is we also looked at the microbiome, and also working with Fred Turek and team, and sequenced them, did both stool and oral microbiome. We could see here the firmicutes to bacteroides ratio, here this color here, increased in flight, which is also a negative indicator of gut health, but it did go back down after flight. And you can see here the colors here, each color on this left is a different species. And you can see the diversity was mostly maintained. But what's really, I think, interesting is about this, we were able to sequence the microbiome in and on Scott, and then compare that to what we saw in the space station. So if we look at the microbes we've been sequencing with Venkat, who's a researcher at JPL, is to see what's on the walls of the space station. And if we look at the T cell repertoire, is essentially what are the T cells responding to in their environment, we saw more of them match the microbes in space when he was in space versus before or after, of course. And so we can actually use all this sequence information to look at what's happening inside the body, how is the immune system responding, what is happening to the microbiome, what are some of the sort of metabolic signatures and stresses of spaceflight, and use this to be a bit of a guide for future missions. So what we ended up saying, at the end of publishing the study is that it's mostly good news. Most of these features across all these different omics did recover. So it's overall good news. But what do we do next? Okay, so what happens now? So one of the things we do is I just included a few studies that we just published back in November of last year, is we included a comparison of Scott to 59 other astronauts data. It was one big package released all at the same time. I was actually very excited because each one of these pictures are ones that Scott took when he was in his mission, and included, each of these as a follow-up study to kind of do deeper dives on some of the data that I just showed you for the year-long mission. And so what we're doing now is actually adding in additional other astronauts out to the year 2033 to look at clonal hematopoiesis as well as other molecular changes that are happening. Here's one of our other missions that was just selected. And you can see we really want to put this in the context of better monitoring and more rapid monitoring during flight. So in particular, one of the other questions come up is, can't we just sequence in space? So we are also a member of this mission, which actually helped get sequencing working for the first time in space with Kate Rubins, who is now currently back on the space station. And we showed this with Sarah Casper-Wallace and Aaron Burton, that you could actually sequence in space successfully, and this is now being done routinely. We also showed that you can get space epigenetics. So you can actually basically get not just DNA, but also modified states of the DNA in space. So I want to take sort of the last 5 to 6 minutes here is to think about, well, okay, this is all interesting and wonderful. We're measuring all these changes. These are dynamic. But how do we go from just measuring and really responding to, can we think about engineering various kinds of protection for the astronauts? We can't just throw them out into the void and say, good luck, of course. We want to think about the best ways we can keep current as well as future astronauts safe and learn as much as we can about this. So one of the studies I alluded to is that we looked at a lot of other astronauts to see what are the other things we can measure that show the damage to their body. One of them is just the amount of 8-oxoguanosine that is being present in urine. You can see here, this is the days before launch or flight day 1530 onward towards the return after the mission. And we can see across all these astronauts a consistent feature of the damage is you can see the 8-oxyguanosine, which again is a damaged DNA base. It's being excreted out of the body. It's literally a molecular readout of how much DNA is being damaged consistently during the mission, significantly, of course, from before or after the flight. And this indicates, even in a low Earth orbit, relatively protected space within the Van Allen belts, it's not good. You're seeing, the damaged bases just flying out of the human body. And that was just, for example, at 6-month missions, or you can see here Scott Kelly's mission, he had relatively, moderate amounts of radiation, 146 millisieverts, compared to, say, a CAT scan, or say the Apollo missions, or Skylab, or even if you're at 1 year on the Mars surface. If you go to a 3-year-long Mars mission, there and back, you're looking at 8 or 9 times the amount of radiation of what Scott observed in that year-long mission. And so this really begs the question of what else can we do? Of course, there's physical protection, pharmacological protection, but what else could we do and learn from to protect astronauts? For example, could we genetically protect astronauts and actually give them, armor that would go on the inside of them? And really begs big questions. Should we do such a thing? Could you think about ways to engineer human cells that would be safe? We do know the way that you shouldn't do this, for example, is to think about making CRISPR embryos and then releasing them onto the world and doing it in secret. This is definitely the way you shouldn't do it. And this has also led to work that with the GP Right Consortium and other collaborators to say we want to have all the methods and protocols and engineering approaches for biology be very open and public and transparent. And this seems like a kind of crazy idea to engineer a new human genome from scratch or to modify human genomes, but it is actually already widely applied, particularly in CAR T therapies and also immuno-oncology. This is us visiting one of the clinical sites doing different CAR T therapies in Shenzhen, China. And you can actually see here, if you're not familiar with CAR T, you can actually take T cells from a patient, engineer those cells so they include a new chimeric antigen receptor, or CAR. You put that on the T cell so it actually can then grow this up, and you infuse it back into the patient. And then the T cells have an exquisite targeting and homing system that will go after very particular cancer cells if all goes well. And so this, again, is really extraordinary technology, extraordinary therapeutic work, really exciting times. And you might think, well, is this just a rare event? Or I heard about Novartis doing this. How often does this occur? It actually is very common. In just the last 3 years alone before this year, the number of trials that are using CRISPR, zinc finger nucleases, or talented are all gene editing systems that are running clinical trials. We just published this last year, has never been greater. There's now more and more trials coming every year that are deploying these and using them inside patients. And so this idea that genome engineering or T cell modification or immune system modification is somehow in the future is just not at all true. It's already being widely utilized in at this point, thousands and thousands of patients, but it is early days. But we've been inspired by the tardigrades that fell a bit onto the surface of the moon, because there had been some papers that showed that tardigrades actually have a protein called DSUP, which actually can be transferred to cultures of human cells almost just temporarily. But we wanted to see if we could actually make this more permanent. So actually, could we actually have a stable lentiviral transduction of cells and get it to reduce the 8-oxyguanosine? And indeed, we can see even at 0 gray, it actually is working to reduce the essentially damage. And consistently, whenever you compare DSUP versus HEC or DSUP versus the vector, essentially we're able to show that a consistent reduction of 8-oxyguanosine. Once you get to 16 gray or 4 gray, it does get a bit harder, it doesn't work as well, but this paper is in review. We can actually show that you can, engineer human cells to contain some of the benefits of tardigrades and give them radiation protection. And so that's one way. But if you're, still thinking, well, I don't know if I'd want to modify my cells permanently, there's other ways that we're actually deploying as radiosensitive basically inducers of cell expression. This is actually a DARPA program as well, where you can actually turn on and off genes using CRISPR methods for the genome. You can also do that for the epigenome. So you can temporarily activate or turn off different genes with their targets using a guide RNA, as well as a DCAS9 and say a TET or a histone acetyltransferase. So you can actually think about the genome as a playground for CRISPR, as well as the epigenome, just turning things on and off just when you need them. For example, if you're going to Mars. And so this actually, is the basis for a lot of ideas that I've just recently thought about. And think about, if we look ahead and think about all the different technologies for genetics and epigenetics, cellular engineering, transplantation, developmental biology. This is a history of everything that's been done up till this year. You can see all these things have happened already. But if you look ahead, I think we'll actually get more protected genomes. We might have more embryo engineering. We might see, therapeutic in vivo transdifferentiation. I think we'll have more people sequenced, of course, on Earth. And actually just finished a book that's just coming out in a couple months that details what I think will happen for the next 500 years. Essentially, we'll have, engineered organelles. We'll have a bit more planetary liberty, cellular as well as planetary liberty. I think the question is, could you engineer humans so they can survive on more than one planet and actually make it so they have, greater planetary liberty rather than just being really, I think, stuck on one planet and having a very difficult time getting anywhere else. And so I think that we've proposed, in this book, a variety of technologies that would make that possible. And this is kind of a preview of what's in the inside of this book that's coming out in a couple months. But my hope is that we would begin to engineer organelles as well as cells. You might have genetic editing that would happen for the whole body. And eventually, if we get really good at sort of epigenetic CRISPR, you could do it just temporarily, not just for occupation, for survival, for example, working on Mars, but could even be recreational at some point and allow you to maybe survive potentially like on Titan or somewhere very far away. This is very, very much in the future, but it's not necessarily impossible based on what we've seen. And then eventually we learn about different evolutionary states of humans that are on different planets. I can almost guarantee they can and will evolve as all life does. And so this is a bit of a shameless plug. It's coming out in April, so I'm very excited that's coming out, though it's been a labor of love for the past few years, and very excited to tell you about it today. It's also worth noting, it's kind of funky that Amazon has a strange algorithm, is that it thinks if you want to buy my book, you should buy Peace with Sweets. I don't really know what that means, but it's just interesting to see that. And I'm really excited for the future. We can think about someone climbing along a canyon here of Mars, looking around the surface, and then looking back up towards the sky. They might see that Earth looks like this, essentially 180 million kilometers away, and they could look back at their original blue pale dot. So I want to thank the twins PIs who made this possible. It was really a big team effort for that mission, as well as other collaborators for the other missions. Thanks, of course, Mark and Scott Kelly, as well as the other members of the expeditions who made this possible. Thanks to everyone in the lab who was really inspiring and awesome. Of course, thanks to funding from NASA, the NIH, World Quant, and other foundations, and Gates Foundation. And thanks to everyone in the lab, and also Cornell collaborators, as well as NASA collaborators, as well as thanks to Kate, who's back on the ISS right now. And thanks, of course, to the patients and their families, and Scott and Mark here as well. And I'm happy to take your questions, and thank you so much for your time.
Well, thank you very much. That was both scientific, but how cool as well. That's amazing. We're going to have lots of questions. We'll ask you one question now, and then we'll move on. But
Great.
First of all, I'll make it a two-part question. Did I see before that you can transfer, you can cool your blood from the station down to Earth? And then did I hear also that you've got a DNA sequencer as well in the International?
That's right. Yeah, it's called the MinION sequencer. It's now actually standard hardware. It's a flight validated hardware. So that we got that up and working in 2016, and now multiple missions are now deploying it and using it for sequencing microbes, sequencing TCR receptors in blood of astronauts. So yep, that's now standard sequencer. So actually, if you propose to NASA, I'd like to sequence something in space, they just say, okay, great. How many flow cells do you need?
And then the blood before was just sort of, was it stored, or did you jettison it down to Earth then?
It usually was spun down. There is a centrifuge on the space station, and then it would be frozen. But there have been other missions where you've done a blood draw and then put it in the Soyuz capsule and brought back to Earth, and then brought over to Houston. So it's actually, you can do it either way. You can freeze it down for later, or you can actually, really have a fresh blood draw and brought back to space and get in your hands in about 36 hours.
That's not bad, 36 hours, is it?
Yeah, it's not so bad.
Not really, no. Okay, well, thanks a lot, and do stay with us. Our next talk is equally exciting. It's by Dr. Michael Waltemathe. He's a senior lecturer at the Ruhr University of Bacal in Germany. Michael is also the co-principal investigator for interstellar exploration at the Interdisciplinary Center for Interstellar Exploration at the University of California at Santa Barbara. He also serves as an officer for the Astrosociology Research Institute. Besides the anthology he co-edited, which was called Touching the Face of the Cosmos, on the intersection of spaceflight and religion, his publications include a monograph on computer games and religion, and several articles on space exploration, ethics, science, technology, and religion within an interdisciplinary perspective. So it's my great pleasure to welcome you, Michael, for a fantastic talk. So over to you.
Thank you so much for the invitation, and thank you for the pleasure of talking to you. I was really looking forward to that, and my brain is still going in circles around the idea of planetary liberty that we will give to humans in the future. I'm going to share my screen, and you will see that what I'm going to present to you is going to be something completely different. I was a chemist, and now I'm a theologian by trade, and I'm going to talk to you about the cultural and ethical aspects of our human future in space, and as it's our human future, I'm also going to include non-human life and just give you a very broad overview of the things we might want to consider when we think about our future in space. Because if we want to go out there into what Buzz Aldrin called, sorry, magnificent desolation, we should be well aware that it is completely different from what we have here on our home planet. And I'm showing this to you because that image, or rather this image, caused social changes on Earth. That image is called Earthrise. It is the image that, for instance, Frank White, who coined the term, relates to when they talk about the so-called overview effect, the effect that astronauts feel when they look at the Earth from the outside. And being a theologian, pardon me for saying that this is also the cause for a little religious incident in 1968 when Apollo 8 flew around the moon, was orbiting the moon, and the astronauts looked back at the Earth. They read from the Christian or Jewish Bible. They read the first few sentences, the story of creation, and thereby, and that's a theologian's point of view, describing Earth as creation, but on the other hand, causing this incident, which is really fascinating from several angles. Couple days later, the American Atheist Association sued NASA for using taxpayers' dollars to send religious messages from Earth, and that court case went all the way up to the Supreme Court. And the fascinating thing about it is the Supreme Court threw it out for lack of jurisdiction in lunar orbit. So when you think about that, the Supreme Court then declared themselves not having jurisdiction about lunar orbit, but now again we're talking about space exploration, where nation states suddenly think about claiming jurisdiction over celestial bodies, which is a really interesting phenomenon, and maybe a religious thing already solved that problem for the future of humans in space. I just want to point that out because I find that's a very interesting thing, and that has had effects on space exploration ever since. But what stayed with us from that moment is this view of our Earth from the outside. And I do apologize for the quality of this image, but that outside look, that overview effect changed society. It gave new power to the ecological movement. It was one of the main reasons for the ecological movement, and it also is something that human astronauts experience every time they go out into space. You could, when you look at the data that is being sent back from the International Space Station since this module Cupola has been added to the ISS, the number of images being taken off Earth by astronauts has largely increased. Astronauts spend more time looking out at the Earth, and astronauts or astronaut tourists even shell out a lot of money to have that experience. Look at this. This is Anousheh Ansari, the first female space tourist. Actually, I think the only female space tourist so far. So she looks out at the Earth from the outside, and all the things that may seem big and impossible on Earth now seem possible. We can do this peace on Earth, no problem. And you all know how hard and impossible peace on Earth really is, but the overview effect sort of gives you the outside perspective. Richard Garriott, British space tourist, he has done it all, right? Skydiving, mountain climbing. He even dove on the Titanic and he went to Antarctica. And then he goes out into space, and this is the moment that changes his life. And that is because humans, for whatever reason, seem to be taken by the view of our planet from the outside. So this is our cradle, right? This is the place where we live now. This is spaceship Earth. And what I'm showing you right now is an artist's concept of a temple on the rim of Shackleton Crater on the south pole of the moon. That is a very sexy spot to go to because you have a lot of sunlight there. If you put up your solar cells there, you will have energy. You can find water there. So this is one of the spots you want to go to. But Jorge Manes Rubio chose that for another reason. He says, if we build that temple there, his concept of the temple allows humans only to see the Earth for part of the time, playing with the idea that this vital look at Earth that is so important for astronauts is not possible all the time from that spot on the moon. So maybe isolating humans from our home planet. And this is the idea I want to give to you. What does it mean to go away from our home planet, maybe to other planets, and only being able to see Earth as it was shown in that image that Chris just showed, this very, very tiny pale blue dot far out on the horizon. So what you see here is is it called Starship? The BFR, the Big Falcon rocket, the rocket that Elon Musk thinks is going to take 150 people on trips to Mars, right? This is the idea behind it. Selling trips to Mars for the price of a medium family, a single family home, 250,000 US dollars, and taking 100 or 150 people on that spaceship to Mars. Now think about that. They will be on that ship for 6 months, right? It's a rocket ship. It's not going to be big, 100 people on there. And everything that society has to offer us down here on Earth will happen there. Everything that is happening between humans here on Earth will happen there. And when you compare that, for instance, to a cruise ship on Earth, you know that the relation between crew and passengers on a cruise ship is very, very good. There's a lot of crew to look after a lot of passengers, right? It's less than 1-to-1, but there's a lot of crew for passengers. Those will not be on that Starship, or maybe you need to put them on that Starship because nobody has ever considered the effects of such a long journey of 100 untrained people into outer space. Now, Chris has told us about radiation dangers, and we've heard about other things. There's also, and that's been alluded to in the introduction, there's also the problem of space sickness. And that's why astronaut crews are highly trained, and crew composition has been something that NASA and other space agencies have talked about quite a bit. And when you read books by, for instance, Mike Mullane, who describes the era when the first female astronauts came into the astronaut corps, everybody expected it to be completely different. It was not, but people thought there were going to be problems. People thought there were going to be changes, and of course there have been changes, but not vast, not fundamental changes. But voyages like this have never happened. They are long, and they take untrained people, space tourists, and that is an interesting thing that I don't want to go into depth here. I just want to point out that could be a problem. Now, there's one instance or one case that we can rely on when we talk about things like this, and this is sort of a weird comparison, because what I'm talking about now is the Mars One project, and from the beginning it was pretty clear for the people who looked at it from the outside that this thing was literally not going to fly. It was the idea of getting people to fly to Mars on one-way trips, paid for by television rights, and basically having a Big Brother show on the surface of Mars. So the idea was, and this was a Dutch television company that stood behind it, send people to the surface of Mars, pay for the trip by television rights, let them live there, and let them die on the surface of Mars and televise it back to Earth. Now, it's the interesting bit. 200,000 people applied for that mission, and they had a huge influx of people, and it was so strong, the influx, that a religious entity, the General Authority of Islamic Affairs and Endowments of the United Arab Emirates, decided to publish a religious ruling, a fatwa, against these trips to Mars. And their argument was, from a religious point of view, Muslims should not apply for that trip because, and I quote from that text, because the risk of dying on Mars is higher as it is on Earth, and Islam is a religion that is interested in lives, in human lives, and so they shouldn't go there because it could be compared to suicide, as the risk of dying is higher, and that is forbidden in Islam. And they had a second argument, which is more interesting even, they said there might be some people who would want to go there to escape God's judgment. And of course, God, as the creator of the whole universe, you just can't escape his judgment by going to Mars. Now, when I first read that as a Christian theologian, that was very interesting because I thought they are making a location argument, as many Christian groups who oppose space exploration do, right? And it sounds like that when you look at it. The risk of dying on Mars is higher than the risk of dying on Earth. Even for a religious person, the risk of dying on Earth is almost 100%. There's not many instances that even religious people believe humans have not died. They all died. Jesus died. So that is not really the argument. And I thought as many Christian groups make that argument that, for instance, if you believe that Christ is coming back to save humans, you should better be here where he's coming back to. This Muslim entity was making the same argument, but maybe they did not, because the United Arab Emirates is now one of the big players of actually going to Mars. Maybe this was all against one-way missions in the first place, because that is the way they thought it and that is the way they wrote it. And that takes me to the other more interesting part of that, escaping God's judgment. If you think about living on Mars and living on Mars, for instance, in this artist's depiction in the future as families with procreation on another planet, that is very, very vital to have that as part of your cultural entity. You can make arguments from religion or from ethics against going to other planets just for reasons that you don't know what the conditions will be, or that you will live outside the scope of what, for instance, God did not gave you as humans. But once you declare that a planet is out there and you can't escape God's judgment, you automatically include that planet within your culture and make it accessible as something that, with that fatwa, is easier for Islam, for instance, as it is for Christianity. Judaism has never really cared about those questions in that regard. They were way more open than the other two religions I just alluded to. Now, why I'm telling you this is because a friend of mine authored a paper on Pew Research data and support for space exploration by religious groups, and especially my religion, so Protestant Christianity in the US, seems to have a bigger problem with space exploration than other religious groups have. And I find it very interesting to talk about ways how to overcome this religious opposition. So when you think about going to Mars with families and living there, you expose them to a different environment. And I was at a conference at the European Space Technology Center in Noordwijk in the Netherlands. And there was a group of art students who gave presentations of research they had done. And one thing struck with almost everybody who looked at it. It was quoted in a lot of talks. There was a poster, and on the poster it said, "After very careful consideration, we have come to the conclusion that life on Mars is shit." And they are basically right. It's very unlivable on Mars. It's not a good environment for humans, and all the dreams of terraforming and so on far out into the future. Maybe Chris is really onto something in changing humans toward that environment. So if you want to go there, if you want to take humans to outer space and maybe even have them procreate there, you need really, really good reasons to go out there because it's very hostile. And this, for instance, could be one of the very good reasons, the destruction of Earth. And even then, you can't go around the religious undertones, right? You can immediately think of, when you look at that, religious people think of the story of the Ark flood mythology. But this person, who is a Republican representative of the first district of Texas in the US Congress, when he thought about that, he thought, and I'm trying to quote this, he said, if this asteroid is going to hit and you send the rocket out that saves humanity, you don't want to save gay people. You don't want to send gay people. Sorry, you don't want to send gay people because they will not procreate and they will not save humanity. Now, behind that is a very, very narrow understanding of what type of people and of what type of social structure you would want to send. And interestingly enough, he's not thinking about that, what is probably most effective. And in the literature on space exploration, especially on going outside of the solar system, you find different concepts. And the one that I find very interesting from an ethical perspective is this, the so-called seed ship. Of course, that's an artist's depiction, and we don't even have the technology yet, right? You still need human females for reproduction. There's nothing like an artificial uterus. But if there were, and if you would send that out, what would that mean? I found a paper online that discusses that from an ethics point of view. You have to ask the question, what is an ethical reason for sending unborn life out into space? And this paper uses a very engineering solution. It basically argues as you have to have a very good reason. The only really good reason can be the possible extinction of all human life on Earth and having humanity live on somewhere out there. So they argue, and the engineering solution to that is you build a dead man switch. You send a ship, it lands on a planet that is possibly habitable, and only if the signal from Earth that says, hey, don't breed, we're still alive, doesn't come, do you start the breeding process out there in space. And from an ethical point of view, or when a theologian talks ethics, that makes it even more problematic. It sounds so easy to say we have this higher value that we put above the problems, and only if humanity is really threatened do we have that ship breed. But that means the necessary condition for those humans to come to life is the extinction of all humanity. So we predetermine them only to live after everybody else is dead. It's one problem. And the other, maybe even bigger problem is what makes them human? Who teaches them to be human? Will that be robots? There can't be live humans then. If anybody were alive out there to wait for the end of humanity, you wouldn't even need the seed ship. So you would need to think about how to make them human in a way that is comparable to us. And maybe that is where this rather hypothetical ethical thought goes together with what Chris was talking about. Maybe those humans we need to send out there won't be the same humans we have now on Earth and here. And I'll try to get this done quickly. I don't want to over use my time. Just again, the flood mythology problem, right? So why send humans? Why not do what they do in these old stories that we rely on? Why not send animals? When you look at depictions of biodiversity, it's always like that. They're cute, they're almost human-like, the ice bear is chilling at the top. So why not send our friends? And that poses other problems that space exploration deals with, or at least asks the same ethical questions, because space exploration tries to reduce life in space. That's a picture of a panel from the International Space Station. There's some mold on that. Usually you don't want unwanted life on the International Space Station. You want to get rid of that. And by trying to do that, by sterilizing and sending only the life you want out there, you sometimes even create life. This is a microscope photo of a Bacillum tursicoccus sinensis. It's a clean room bacterium that has developed or has been found in clean rooms at NASA and the European Space Agency at roughly the same time. So while we try to restrict life during our space missions, we actually create new life inadvertently, which poses all kinds of interesting problems with that. And now just one more thing that is very dear to you, and then I'll let you go, very dear to me, and then I'll let you go. All this plays into the question, if we send stuff out there, what rules do we want for that? And as an example, I'm going to just shortly talk about planetary protection rules. We already have rules for sending Earth life, non-human life, but with human life comes non-human life out into space to other solar system bodies. So we have rules for contaminating other celestial bodies in our solar system, moons and planets. That's called forward contamination. And we have, well, rules for backward contamination. Not really, but we have very strong ideas about that. So the current rule for missions to so-called category 4 sites, those are sites where there is liquid water or warm ice, potential habitats for Earth organisms, define the maximum risk of contamination to be less than 10 to the -4. So if you send a mission to a category 4 site anywhere in the solar system, you have to ensure that the risk of contaminating that habitat, that possible habitat with Earth organisms, is less than 1 to 10,000. And I authored a paper with 2 colleagues from JPL, and we're basically asking, where does that number come from? It's a historical number, it's a can-do number, it comes from sterilizing fruit cans and sterilizing surgical equipment. But why is that number? The rationale behind it is the protection of science. But shouldn't we, if we want to send life out there, think differently than just protection of science? Shouldn't we argue ethically, and shouldn't we question that number for scientific reasons, because we know more about how life survives, how life proliferates, but also for reasons of ethics? You could ask, why that number? Why not any of those numbers that are in the list? Why just have the risk at 1 in 10,000 and not 1 in 133, like the admission to RuPaul's Drag Race in 2017, or 1 in 11,000, the yearly death from the risk of dying from assault with a gun in the United States? In Europe, it's a little less. So why that number, and not why not the others? And you would carefully need to consider ethical reasons for that. For instance, the value of extraterrestrial life compared to our life, the risk of extinguishing a whole ecosystem by inadvertently introducing an organism that kills it all, or maybe the superiority of Earth life over all other life, which is not a very good argument in my opinion. But on the other hand, we have no other occurrence of life in our solar system yet. So maybe we're the only ones, and we need to go out there. But you can even argue against that by saying we should have planetary park systems that protect the natural beauty of other planets as they are now, and not send our ecosystem out there. And that almost sounds like a religious argument, saying they were made the way they are and we should keep them that way. And with that, I'll leave you to it. And thank you very much for listening to me.
Great.
Well, thank you, Michael. I think you blew us away. I can look at the chat and I see so many people giving you compliments. I hardly have ever seen it this way.
Oh, I see you. I see you again. Something has frozen here. Oh, there you are.
Okay. There are quite a few questions. Just ask you one question and then we come back to you after Kira's talk for the general Q&A, of course. So please stick around. But I'll ask you one question now, which is intriguing. There are many different cultures and religions on Earth, obviously. And we could expect that when there's a good proportion of us going to space, that this will stay the same. The representation of these different cultures and religions. So pragmatically speaking, how do you deal with the rules, such as Jews observing Sabbath? When is it Saturday? When you're spinning around the Earth or going into space? And what about Muslims looking for Mecca when they're on Mars? So I mean, it's just all these practicalities that are common for these world religions. How are you going to deal with those rules?
there's actually two parts of your question, and one has become more and more interesting for me. There's this dichotomy between we have a very diverse set of cultures on Earth, and at least I perceive it that way, the need to have a very uniform motivation for space exploration. And that is, it's not necessarily in conflict, but it's interesting. I'm just working on another paper on that. It's very interesting to see how those might collide in the future. And we should think about that. And the other thing is the practical thing. There's actually been another religious ruling on which direction do you pray when you're in low Earth orbit, right? And for Muslims who are on the International Space Station, for instance, and that's actually from the tradition of Islam. So if you are in Mecca, you're staying in front of the Kaaba, you pray to that. If you are outside of Mecca on Earth, you pray in the direction of Mecca, the geographical projection. If you don't know where you are, you pray in your heart to God, not to any other God, but to Allah, and then the direction doesn't really matter. And the astronaut who actually had that ruling written for him, when he was asked by the press, where do you pray? He said, Allah wants adoration, not acrobatics, and that's what I'm going to give him, which is a really, really good answer. And so the Sabbath thing, just very shortly, one Earth orbit on the ISS is 90 minutes. So 7 times 90 minutes is 7 sunrise and sunsets. So the Sabbath is being held when the sun sets on the 6th day, and before it, until the first stars appear in the night sky on the 7th. So you would have to work for 6 times 90 minutes and then make a break for 90. That wouldn't work. But luckily enough, there's a religious institute for space halacha that deals with that question, and they could relate to a ruling for Jewish submariners who would stay under the surface for a month and would need something to hold on to. And the ruling is easy. God created the Earth with a 24-hour rotation. Why should you change that time frame? And so you just obey the same rule, and the time zone you focus on is the one from the launch site. So very pragmatic, but only, and that's interesting, only for low Earth orbit, right? There's a science fiction story actually about Muslims praying on the moon, and to get the angle right, they build a little contraption that you get the angle right to pray towards Earth, which is just an extension of that rule. But going further out, it could make it more difficult, especially when you think about pilgrimages. You would need to go back to those places, and you possibly couldn't. It's very interesting, but far out in the future.
Yeah. Thank you very much. Thank you again.
Thank you.
It's now my pleasure to introduce to you our last speaker, Dr. Kira Bacal. She was born in the United States, but she's coming to us from Auckland, New Zealand. She received her bachelor's in mathematics and biology from Amherst College in Massachusetts. She attended Bacal College of Medicine, where she earned a PhD in molecular physiology and biophysics, and a medical degree, as well as a master's in public health from the University of Texas. She trained in both emergency medicine and aerospace medicine, a great combination, and she was in the US Air Force Reserves. She worked at NASA Johnson Space Center in the Office of Space Medicine's Medical Informatics and Healthcare Systems, designing medical systems for use in space. Dr. Bacal also worked in health policy in the US Senate and at Ohio University before immigrating to New Zealand in 2007. She now serves as the deputy head of one of New Zealand's 2 medical programs, as well as working for a medical assistance company. And her title is the best after the other 2 speakers. It is, but now what? Where do we go from here?
Thank you very much, Jacques. And again, thank you all for both the invitation to come here today and the opportunity to speak, the attention of participants. I will say that I find it a very daunting task to follow such fascinating and thought-provoking presentations by Chris and Michael. So, no pressure, no pressure. I will also mention, Jacques, that you are not alone on my flight on the aptly named Vomit Comet, which of course, as you recall, is the NASA plane that does the parabolic flight. So you get essentially 30 seconds of microgravity followed by 30 seconds of 2Gs. I put the vomit in Vomit Comet, so you're not alone, my friend. What I'd like to do in the next few minutes, and this will be a much shorter talk than my predecessors, is to summarize some of what we talked about and perhaps refocus it a bit more on the concept of reproduction and procreation specifically within the space environment. And I'll do that against the backdrop, as you heard, of my former work developing medical systems for use in space, and that was low Earth orbit, Moon, Mars, and beyond. I think the main thing to remember, of course, is that the real question for many of us is whether development, and by that I mean from fertilization through formation of viable gametes in the next generation, can actually occur in the extraterrestrial environment. And the reason that this is critical is because this is the underpinning of the goal of human exploration. I reflect back on Michael's recent slide showing the planet killer event. If the goal is to get out to preserve our species from that extinction event, then presumably we need to make sure that we can procreate, whether with Chris's proposed machinations or indeed just because life will find a way, to paraphrase Michael Crichton's Jurassic Park. All human life has developed within the same parameters of radiation, gravity, atmospheric composition, and a very narrow band of ambient pressure and temperature. And our question, therefore, is when this is not maintained, either in a artificial space station environment, a interplanetary vessel, or on the surface of an extraterrestrial planet, planetoid, asteroid, moon, etc., can these processes either successfully proceed or be made to successfully proceed? And I look forward to reading Chris's book for some of the ways that we might intervene in that. In the interest of time, I am going to avoid talking about the psychosocial aspects associated with procreation, human procreation specifically. And I'm also going to avoid the mechanics of how the sex act might work in a gravity less than a full G. For those of you who are interested or are troubled by insomnia, I will refer you to the article I wrote back in, January of 2009, which is on Medscape Internal Medicine, and it's available for free. So there's my shameless plug, Chris, you're not alone. That said, I don't want you to think that my not mentioning it in this forum suggests that the psychosocial aspects should be underestimated. This also is obviously goes along with what Michael was saying around the ethical implications of trying to regulate human behavior in any way, and particularly if you're trying to curtail or regulate that behavior. Instead, what I'd like to do is review some of the physiological issues that are affected by spaceflight and its attendant changes to everything. From, as we've heard, gravity, radiation, noise, vibration, isolation, circadian rhythm changes. And yes, Jacques, your question and Michael's response about how do we manage the circadian rhythms from a religious institution standpoint. I just kept flashing on the old joke of the Jewish astronaut saying, every 90 minutes, off with the tefillin, on with the tefillin, off with the tefillin. So this is why the Halachic Center obviously had to make its ruling. The other point I'd like to make is that the challenge of studying these should not be underestimated, because there is some very good evidence out there to suggest that the impact of these numerous changes, one might almost say the myriad of changes that occur in a spaceflight environment, do not happen in isolation from each other, and that the impact that these complex biological systems experience is synergistically as opposed to additively felt in some cases, and in other cases there may be oppositional directions. So as I talk about some of these changes and their implications, again, to reproductive biology, I'd like you to think about how, if you are a researcher, you might look to study these. If you're not a researcher, what questions you might have and how you might look at exploring these. Our technology is constantly improving. We were hearing from Chris that there's a DNA sequencer aboard the International Space Station now. And yet we need to try to expand our understanding so as we heard from Chris, we can explore countermeasures. My particular background was in cardiovascular changes when I was back in the Stone Age. When I was a student, we were entering the shuttle era, and for the first time, instead of our astronauts simply hanging on as their capsules splashed down and the US Navy went and fished them out, literally in the case of Gus Grissom and the Liberty Bell, we were seeing people pilot a craft to a controlled landing. And when we discovered the orthostatic hypotension that accompanies return from exposure, even very brief exposure to microgravity, the problem obviously was if this crew becomes dizzy either during the piloting of the shuttle or during an emergency egress, it's very difficult to escape while you're unconscious. So both research into what was happening and countermeasures as to how to stop it from happening were going on simultaneously. And that was the underpinning of my PhD dissertation, and enough about that. So as we talk now about these effects and how they impact reproductive development, please be thinking in your own minds of how would I study that, or what are some of the problems with studying that. We know that the spaceflight environment affects gene expression. And that means that all aspects of development, from cell structure and function, organ system development, all the way through behaviors, are going to be affected. We also know that there are periods of maximum susceptibility during development, but that these are likely to be different for different organ systems. So when you think of these countermeasures, you have to think in very broad terms. And you have to appreciate that for many of these, the longer the exposure, as well as when it happens, the longer the exposure, the greater the risk of an unwelcome change, such as a fetal abnormality. The literature reports many of these changes. In our first seminar back in October, as Giles was mentioning, Dr. Sedrudin gave a very nice overview and went into some detail on some of these. I'll very briefly just mention some. We know there are changes in testosterone levels and other endocrinological changes. Obviously, you can think of what that's going to mean for a developing fetus, for example. There are changes in sex drive and mating behaviors. We know that there are changes to male fertility. We were hearing earlier from Fathi and his research into sperm motility on the ISS, and therefore changes in fertilization overall. We know embryo survival in many species is decreased with exposure to space flight. There are problems with surviving to the morula or blastocyst stages. There's problems with cleavage and neurulation. We know that space is a teratogenic environment, leading to developmental abnormalities in many species, ranging from bone development, vestibular systems, visual systems, and so on. And we also know, interestingly, that in at least one species, there was an increased number of deaths following live birth after flight. So is there an impact that lingers? We saw from some of Chris's slides that we know that many of the changes occur and then rapidly taper off. But here's some evidence that in reproductive biology, at least, there may be a long shadow. And Michael, I'm trying very hard not to talk about the sins of the father, although I'm very tempted. It's also worth noting that there was a study that showed that neonatal rats that were exposed to space flight at certain times in their development ended up with long-term problems with motor function. So again, if we think back to that slide of the family happily frolicking on Mars, you may have to think about, in the event of space tourism actually taking off to a significant degree, do we need to restrict that travel to either duration or age, not just from the standpoint of the risk that someone takes when going on that, in the same way that many adventure tourism activities restrict age, but from a physiological standpoint of, if you bring your child into a space environment at this particular stage in their development, they will always have vestibular problems or what have you. Or if your pregnant wife travels during this time, the baby will potentially have a long-term impact. So all of this obviously implies that mammalian growth and development can be profoundly affected by the space environment. You've heard lots about radiation. I'm not going to belabor that point much more. Virginia spoke about this very eloquently at the first webinar, but we know that this is, as Fathi mentioned, probably one of the major challenges to exploration outside of low Earth orbit. We know that low Earth orbit itself, as we heard from the twin study, has many impacts, and yet the radiation environment is substantively different as soon as you escape the Van Allen belts and you go into any kind of interplanetary or even from Earth to Moon travel with DNA damage, cell death, chromosomal abnormalities, fetal changes, and so on. As I mentioned before, though, it's not that simple. It never is. And there is good evidence to suggest that there are synergistic effects between, among other things, the radiation and the microgravity, leading to greater damage when both are present than when either one is alone. One theory suggests that the microgravity affects the cell repair mechanism so that the radiation-associated DNA damage cannot be readily addressed. And as I mentioned before, this further suggests that increased exposure leads to increased risk and makes investigation more challenging, because when you research one aspect, you may not be getting the full story. And therefore, any generalizations or mission planning that goes on on the basis of that study may actually misrepresent the actual facts. I'd like to also, because I think many of you, I've seen some of the chats commenting about clinicians and OBGYNs and the like being present. So I'd like to just also point out the question of, what do we do with a pregnant woman? Many of us, either as pregnant women or being faced with pregnant women, say, oh my gosh, that's a problem no matter where we are. But I assure you it would be much greater in space. It is unknown at this time, for obvious reasons, if the adaptive changes of pregnancy are synergistic, additive, or oppositional to the adaptations that the human body makes to adjust its homeostasis to the spaceflight environment. Depending upon which organ system you look at, the answer is probably yes to all of the above. What we do know, certainly, is that both pregnancy on its own and spaceflight on its own create changes in most of the body systems, ranging from gastroenteral, GI system, hyperemesis gravidum, space motion sickness, cardiovascular, mild stomping grounds, where it changes to red cell mass, blood volume, bone mass, and so on. And we also do know some animal model studies that show that duration of labor can increase, birth weight can decrease, and even in rats, at least, postnatal maternal behavior changes. There was also a study that showed that post-flight male rats, so the male rats were flown, they came back, they were then mated with female rats who hadn't flown, And in the offspring of that pairing, the baby rats were seen to have increased abnormalities as well as growth retardation. So all of this obviously suggests that we need to be extremely careful in not only thinking about how the human body itself adapts and what we might need to do to fully functional adults with good physiology, no major health issues, and the intellectual capacity to make their own decisions, but what do we do with pregnant women and indeed, as Michael was mentioning, unborn fetuses. I would also recommend that if you'd like an excellent overview of women's health in space, the previous webinar's presentation by Dr. Jane will provide that to you. As someone who designed medical systems in space as a living, I can promise you hand on heart that the systems are not prepared for a normal pregnancy or childbirth, let alone high risk ones, which almost by definition we would be dealing with. There have been a lot of comments about the fact that astronauts, US astronauts in particular, just because their numbers are greater than our colleagues at other space agencies, the female astronauts often become mothers at an older age. So you have greater maternal age as one risk factor. All of the above developmental issues that I mentioned, contributing to things from ectopic pregnancies, preeclampsia, low birth weight, gestational diabetes, RH incompatibility. Any of these would have an enormous impact for the crew and the mission, with ramification for the mission objectives, team dynamics, morale, evacuation or mission termination, and of course, crew safety. And I think that one of the things we often forget as we talk about this in the abstract is that we need to consider it not only from the standpoint of the individual, but from the standpoint of the crew and the team and the mission, because these are, it's almost more like a military objective or operation than a civilian one. These people are being put, at the moment at least, in a space environment for very specific reasons. And if those missions cannot be achieved, there is an enormous cost to society in terms of dollars spent, but we also know it in terms of the crew safety. And what we've seen with all of the previous tragedies, whether we talk about the loss of cosmonauts on the Solid space station, Challenger, and so on, we have to consider what the threshold, the public threshold and appetite for risk is. And I would argue that it is very, very low in the Western world. This implies enormous things for our commercial space and space tourism industries as well. So given all of the above, it's clear there are many ethical questions around pregnancy in space, including, and Michael may speak to this more, but you know, how do we see if people can reproduce off Earth? Because as I mentioned before, this underpins the entire plan of space exploration as a goal to safeguard humanity. But as any parent will surely tell you, just because you can do something doesn't mean you should do it. And so Chris has talked a lot about what technology is likely to allow us to do in future. And he and Michael have both spoken about the need to have strictures and structures in place to ensure that it is done in an appropriate fashion. I'd also ask you to think about the implications for the mission if someone were to get pregnant during flight, and whether that's on a space station mission currently or on a longer exploration mission like the ones you've heard about to Mars for 3 years, as is currently planned. Again, I'm not trying to talk about could they get pregnant, you know, logistically, could they get pregnant? If they did get pregnant, what's the current NASA plans for it? Could we diagnose pregnancy and so on? The answer is yes to all of the above. But just what are the moral, ethical, and logistical implications if that pregnancy takes place? Do we need to evacuate immediately? Most of the space missions in the near future will have an evacuation of one needs an evacuation of all. And that has enormous implications because the ISS was not designed for unmanned operations. So if you, unlike the earlier near space stations and so on, which were like a vacation home, you can board it up, go home, it's fine, it keeps going around, and then you go back when you want. Instead, the ISS is intended to have somebody in the driver's seat at all times. So if that comes down, you have potentially the risk to the platform as a whole. You lose the entire platform. So I think that then we have implications for what you've just done. You just lost the entire mission. What if the mother says, no, don't bring me back, I'm willing to take the risk? What are her rights as a sentient individual versus those of the unborn child? Of the fetus versus the rest of the crew, if returning, particularly from an exploration mission, entails a great deal of risk. And as I mentioned before, of the fetus or the pregnancy, what are the rights pertaining to the unborn child versus the mission and the social contract that set the crew out? So all of these are interesting. And if we say, okay, fine, we're not going to cancel the mission, then what are the implications of trying to manage a pregnancy and indeed a miscarriage or a live birth? So as any good PowerPoint will do, I will conclude with the fact that clearly future research is indicated. We're all scientists here, we all have to say that. I think that some of the research, as you've heard today, needs to investigate the synergistic effects of these different stressors on living systems, the extent of the teratogenic effects and countermeasures to them. And obviously, as we've heard, the research is needed on virtually every stage: fertilization, implantation, embryogenesis, fetal development, pregnancy, childbirth, prenatal care, postnatal growth and development, sexual maturation, you name it. And the ethics of how do you study that in humans in an appropriate way is obviously a question in its own right. So in summary then, I will say that space environment, obviously, as we all know, has effects on mammalian gene expression, which may have significant ramifications for eventual human procreation. Exposure to microgravity at certain times and for certain durations can lead to long-term abnormalities on organ system development and function. And lastly, I don't want us to sound too speciesist, because it's not just humans we need to think about. We know from animal model that mating and reproduction in many, many species, vertebrates and invertebrates alike, will be problematic. And that has implications for the development of self-sustaining extraterrestrial ecosystems with in situ agricultural production. Thank you very much.
Thank you very much. You've sewn together the first two talks perfectly, and there's so much we can talk about: the policy, the law, which we wanted to talk about. But you mentioned, and again, about the underpinning is of course reproduction, or else why perhaps we're doing this. And the numbers are low, so all the studies, of course, have just been limited to what number. So how important do you think, like, the moon landing is going to be? Is that going to help us, especially with the Mars missions? Are there going to be more experiments going on there as a sort of an interim?
Yeah, I think that there's long been at NASA, the debate about going back to the moon versus going straight out to Mars. I think that I think that there's a very good argument, you can see which side I'm on, that we need to go back to the moon and use it as a test bed to learn more about not only the engineering that will be required, but the biology, the human aspect of it, before we do a Mars one and send people out on essentially a unsurvivable or one-way trip. I think the other thing that's quite interesting, and I have to bring this up, living as I do in New Zealand, we need to also look back at our history, because colonization, whether it's of the moon and Mars, is hardly a new thing for humans to do. We've colonized a lot of places which were very hostile in their own ways over the course of human history. And I think, and we know, looking at Michael's talks about how do you contaminate an environment, invasive species and the like is a huge issue for many of those colonized regions. I also think it's interesting to look at the assumption that we're all making, which is that space missions in future will concern themselves with some of these ethical issues, because we certainly have plenty of examples, both in fiction and in history, where colonization was done by forced conscripts, and there was not a concern about procreation. It was go dig those mines, or just don't come back. Whether we're talking about Australia as a penal colony, or Robert Heinlein's seminal work, The Moon is a Harsh Mistress, which proposed the same fate for the first lunar colony. So it's very interesting, and I think we just have to, hope and pray that we continue to approach it in a very open and multicultural and respectful fashion, and not use it as the new prison labor camps of the 22nd century.
Right. Okay. Thank you very much. We've heard our talks. We've each had a question or two each. Can I invite everyone back, panelists back as well? Okay, they've been waiting a long time. If I just kick off with the first one then, and is Fathi with us? So, of course, you were the first section. Is there any more work to be done? I mean, after the Micro 11, we have some questions here about the capacitation. Was it long enough to do? And does it have to be done in the International Space Center? Because I hear they could be in like biosatellites. Perhaps you could shoot them in space, and they could rotate and come down. Is that anything that can be done?
Yeah, I think all of you above would be more than appropriate to try to address all those important questions that all the speakers have into it. And so I think that more the merrier. I think that there's still little is known about the overall human physiology and how that is impacted by different environment. And, microgravity is one of them. But as Kira mentioned it, I think that we need to have a synergistic approach about all those compounding effects on the overall physiology. And I would correlate with what Chris has been mentioning. I think that you need to have a global perspective and a global view of the human body from a physiological perspective to try to address all those important questions. And so I think not just NASA, but I think all space agencies has to work on all those different topics. And I think luckily we have a lot of talented people that can address those questions. And so you're right. I think doing more on the station is important, and having capabilities like sequencing technology on board the International Space Station is important. Currently, we still, despite the fact that there's a lot of astronauts and, we've been on the station for the past 20 years, it's still very difficult to conduct operation. It's still very difficult to get access to cold storage. It's still very difficult to get samples returning to the ground to do analysis. And so having all those in situ technologies, that would be critical for us to be able to, conduct science like we do in the laboratory. You have an idea, you test the idea, then you get the results, and then you refine your idea and so forth. And so I think all those elements are critical to be able to sustain long duration spaceflight and address all those risks.
Okay, thank you. Jacques, do you want to kick off for the Q&A? A few more questions?
I'll be happy to. There's a question here from Paran Tayebi for Chris Mason. He's thanking you for your talk and is wondering, have you also looked at genes from people that lived in extreme environments for their lives, with the result that their genome changed? For example, people living in the Arctic or in high altitudes. Maybe those genes could be used as well as to make human cells stronger for spaceflight.
Yeah, great question. So there's a list of genes also that we're in the middle of writing a paper with George Church on this, because he's also proposed this for a few years, of really interesting adaptations that are already known for people, for example, deep sea divers in the Philippines, as well as like people, Sherpas in high altitudes. There are genes that have been associated with some of these sort of interesting adaptations. We could almost call like superpowers that are present in normal people today. And, I think, as I alluded to, there's ways you can stably, transfect human cells and have the gene be integrated. But that is something that I think both just socially and structurally for the genome, people always be a little bit hesitant about. But what I think is much more palatable and also much more controllable is just Most of these genes are already present in the genome. You can change their function or their activity, or if temporary actions that would keep some of the genes present. So I think the sort of malleability of some of the genetic features is something that was really unimaginable 15, 20 years ago, because we didn't know enough about epigenetic regulation, but now is actually already being done clinically. For beta thalassemia trials, one of my favorite examples is for people who can't make adult hemoglobin that well. If you recall, when you were all fetuses, we had fetal hemoglobin, which actually binds oxygen even better. And they just reactivate the gene, which has been silent ever since you left the womb. And so we have within us all these genes and gene segments that could maybe be reappropriated. Another example is vitamin C. It exists as a pseudogene. All of us are dry nose primates called haplorhines, and there's all the wet nose primates like aye-ayes and other primates out there that they still make their own vitamin C. So they don't have to get it from their diet, they can make it. So there's all these little nuggets in our genome of features that we either used to have or we could have or we could tweak a little bit. And so I think they'd all be fair game for sure.
Yeah, great. Another question that came up early, that's really probably for most of you in the panel, is it goes back to our field, which is when the first mammalian embryos were frozen in the 1970s. Since all molecular interaction stops, we don't have to worry that much about normal chemistry at those liquid nitrogen temperatures. The worry was that if you keep embryos frozen for a long, long time, background radiation would be a concern. And so a few papers were published in the 70s about this. And one that comes to mind is the one by Mary Lyon, and David Whittingham is an author on this as well, who was the first to freeze the mouse embryo. They published a few papers measuring background radiation, but also imitating background radiation on Earth for about 10,000 years, maybe 30,000 years, thousands of years. And the question is, should that be revisited? Because those experiments can be done, I guess. It had no effect at the time on embryos. Of course, the endpoint was nothing like what you, Chris, are presenting. It was on survival, morphology, development in vitro of these embryos that were thawed and then transferred to recipient foster females. And the thought was that they actually developed well and that they go to birth and seemed to be looking like mice and behaving like mice. But it was not on a molecular level as far as I remember. So if you compare background radiation on the planet, on the surface of the planet, with that in space, like on the ISS, can you at all, what is the ratio? What's the ratio? What is the effect? How would you have to imitate what the effect is? Can you? You're muted, Dr. Kira.
Oh, sorry, I didn't know I was allowed to answer. Yeah, as we've heard, low Earth orbit is still considered a radiation worker's environment, and it has greater radiation than the surface of the Earth, as Chris nicely demonstrated in some of his work with the twins, and I think Virginia mentioned in the previous webinar. The big problem, though, is that as soon as you leave low Earth orbit, which is, again, where, in my view, the money question is what happens outside of the planet, and I include low Earth orbit as being within Earth, it's a substantively different radiation environment. So it's not like apples and oranges. It's like apples and giant sequoia redwood trees. I mean, they're like two completely different things. And so I think you have to be aware that when we're talking about ionized iron, for example, these enormous nuclei that are racketing through space and the galactic cosmic radiation, what they can do, it's kind of like, driving a semi truck through a building in terms of the impact they can have on the DNA. So it's very challenging even to replicate that on Earth and to make sure that we're getting it right. So I think that would be very problematic. That my opinion. I'd be interested if Virginia or Chris had another comment.
I mean, I'm happy to jump in and provide some numbers and values to the audience which might not be familiar with the radiation exposure in low Earth orbit or on the moon or on Mars. So if you measure the background radiation on Earth, let's say, like, for example, Houston, which is the main places where all the astronauts are located, it is roughly like 2 millisieverts per year, actually 1 millisievert per year. And if you go and perform a transatlantic flight, it will be 0.1 millisieverts per flight. So roughly that kind of give you a value. But what is more striking and more important is like if you go to your doctor and get an X-ray of your chest, roughly you will have a dose which is like probably 1.1 millisieverts per exposure. However, if you're in space and in low Earth orbit, you will be receiving 5 to 10 times that exposure per day. So going back, you go to the medical doctor, you go for an X-ray of your chest, it's 0.1 millisievert per exposure. On the International Space Station, depending on the solar cycle, you will be receiving 5 to 10 X-ray doses per day. And so as you can see, it's a kind of large amount. But what was Kira alluding into is that you have different type of particles, and some particles are more damageful than others. From an occupational point of view, currently the astronauts are the only humans, I guess, where that have, from an occupational perspective, receiving the largest amount of radiation. If you are a radiation worker, the limit is 50 millisieverts per year. However, for the astronauts, is 500 millisieverts. So it's 10 times the dose from the radiation worker. And so Chris alluded into it that, you know, if you look at a mission to Mars, roughly based on data from some of the spacecraft which have been orbiting around the planet, that would probably, you know, lead to a total dose of 1.2 sievert. And currently the limits from lifetime limits from the astronauts is between 1 and 4 sieverts for an astronaut. And so as you can see, certainly from a radiation perspective, I mean, the doses are very large. And certainly when we escape the Van Allen belts and the Earth protecting atmosphere and geomagnetic sphere, certainly you're exposing yourself to a very, very large amount of radiation.
All right, thank you.
I just also point out that we can't look at the trip to Mars in a vacuum. It's very unlikely that an astronaut would be selected to go to Mars, or most of the astronauts would be selected to go to Mars as their first trip in space. So as Fathi's pointing out, they're already getting high doses, and then you're stacking it. There was a big fight when I was at NASA because there was some person who tried to argue for a particular astronaut that we needed to discriminate between radiation exposure that they got for their personal health issues and radiation exposure that they got professionally. And I was pointing out that if the cells don't discriminate, then we shouldn't either. The argument sometimes. Anyway, so I think that that's another point to raise, is that it's not just, wow, that's a really big whack. It's actually going to be on top of the already big whack.
Yeah. So what about countermeasures for this? Somebody suggested, maybe this was the last session, to use graphene foil as a way to wrap humans and objects and maybe the entire spaceship. Is there any other suggestions out there?
Look, there's lots of suggestions out there. This is, I mean, you know, I think, was it Chris who was talking about, you know, you can be a lunar architect. There certainly is a whole branch of engineering and radiation around radiation protection, and that involves also, Chris was talking about, you know, internal engineering to outside options, to from force fields to material science, storm cellars, what you wear. There's not yet been a brilliant solution to fix it, and that's why people, you know, the next X Prize perhaps. One of the challenges, of course, is that when you're talking about these enormous heavy metal nuclei that are coming through, you have to worry about background radiation. So you wrap something to shield them, the radiation hits into it, and it's like those little balls where you hit the first ball and it next knocked back, and the next one flops out and it won't. So you don't want to create radiation inside the shield from the radiation on the outside. So it's not an easy mix.
No.
Sure. Is there anything that, I think that Virginia wanted to add something there? Was that?
So we often, the first thing we think of with radiation protection is shielding, which we don't have a good answer there yet. The next thing that people think of is some kind of drug, some kind of magic pill to help us recover or repair or protect us from radiation. And unfortunately, we don't have that either. We do know that radiation has really complex effects on the human body, on our physiology, particularly chronic radiation, I think. Our bodies begin a series of biochemical reactions in response to radiation that trigger downstream effects and continue at the same time that more radiation is being absorbed. Very complicated. How to target a drug against that, we cannot even imagine right now, although lots of people are trying lots of different approaches. There are literally hundreds, at least, of compounds that are being actively tested as either mitigators of radiation damage or protectants against radiation damage. And it could be that we figure out something useful in the not too distant future.
Sure. And in fact, one of the interesting things when I talk to people, when we talk about this session, is one of our former speakers, who's Francesca Klinger from the SIA Italian group. They're in fact working on a material, I think, as well with the Italian government. But if I stay with you a little bit, Virginia, I think it's a 3-year round trip, and we haven't really talked about female physiology. If you could tell us something about that, and bringing in Chris into the equation, are they going to perhaps do a twin study where they're both female?
Oh, wouldn't that be wonderful? Yes, indeed, indeed. I don't think any space agencies are actively recruiting twins at this point, but certainly when they happen, we'll take advantage of them, right, Chris?
100%, yeah. And I think there is a real disparity between the number of obviously male versus female astronauts that have gone up, and females so far seem to have a little bit less incidence of SANS, the spaceflight associated neuroocular syndrome, where they have damage to their eyes and retina and can't see as well. So there's often some jokes at NASA that most of the first mission to Mars might be more females than males potentially, but it's too early to tell. The numbers are too low, but yeah, I think obviously we need to understand as a function of age and sex all these factors.
There are also some advantages when it comes to body size and resources needed to keep people alive in a closed environmental system. You know, smaller people require less oxygen, less food, less water. And for that reason, people sometimes consider that more females would be advantageous.
And I think that it was thought once upon a time to have an all-female crew, I think, go to Mars. I saw that to the headlines. But if I bring in Chris, and then I'll get to Michael in a minute. What about mitochondrial DNA? Isn't there a lot of cell-free DNA, and there's damage in that sense?
Yeah, that's what we've been saying. So we did a lot of cell-free DNA profiling. You can do it from, we now have multiple grants to do it from urine as well as from plasma. We can see a lot of damage to the mitochondria, and what's, you know, the powerhouse of our cells really shows a lot of stress in spaceflight and is even ejected into the serum. So these were, again, surprising features that you see, and you see them in stressful situations on Earth. You can even, there was a study at Columbia that actually did blood draws of people before and after they gave a public speech. And a good number of people actually get so stressed when they give a talk in public, they have more mitochondria ejected into their bloodstream from lymphocytes. So, you know, a lot of it's just the stress of the body and how it responds is what's called an interferogenic response, where the body, when it's unclear what to do, it tries to prime it almost as if it's being attacked for an infection as kind of a safeguard. But, you know, there are just weird things that when you start to look at a high resolution, you'll see strange things in how the body responds to space. But we also want to always put this in the context of what do we know about just normal physiological stress. And so, you know, I think every organism though is going to be required for the view. And so there's some microbial species that make small molecules that seem to be radioprotective. They can help with nutrition. We should think about, you know, not just the human cells, but the microbial cells. In fact, any human geneticist today, if you only look at human DNA, you are a bad human geneticist. You have to consider all organisms that contribute towards human physiology, which is more than human cells. So I think people are getting more systems thinking about their view of biology and space medicine, which is good.
Great. And Michael, should we be tampering around with DNA? You know, or from an ethical point, is that something that we should be doing, or should we just keeping ourselves pure?
Is thank you for only giving me the easy ones. Yeah, sure,
Yeah.
why not? I mean, so just one remark to Chris. I'm pretty sure that wasn't talking. That was just the fear of getting the needle. That's good. That research that you just alluded to. No, but Giles, I mean, we've been doing that for centuries now. The work on DNA, not the way we do it now, but I'm pretty certain I'm eating tomatoes that have not been that way before we crossbred certain plants. And also, you could argue that nature has been doing that. And if you want a theological argument, you could of course argue, no, you shouldn't do that because it has been created this way, but we all know that it has not been created this way. But if you believe that it has changed, right? And you could also make the argument that once if you really want to go back to this relation, we've been given the tools to do that. So why not use them? And maybe the only problem is when you really, so if we would start creating a whole universe, then maybe we would get in what's the word? Oh boy, I'm missing words. I'm sorry, my English is I'm missing English words in it doesn't matter. Then you would get in trouble. But as long as you work within that what we live in, creation, the world, the universe, and work with the tools you have, I think you can make a pretty good argument to just use those tools. And in that regard, there was a question in the chat. Hang on, I'm trying to find it. It was pretty early on. It was about turning the overview effect upside down, basically, and saying, listen, the more doesn't science, this is my translation of the question, doesn't science cause something that is similar to the overview effect? Right? We don't have to look at the Earth from the outside. The more you learn in science, the more fantastic, the more fascinated you can be. And I would totally agree with that. And even from a theological point of view, and that's one of the questions I always get asked, I don't think you can find God through science, and I don't think anybody expects that. But if you think, or if you are of the belief that this was made by somebody, then why not use science to understand the mechanics, right? Not find the person behind it. That's a Christian term of putting it, but try to understand the mechanics with the high risk of maybe finding out that there is nothing behind it. But that's a risk you got to take, and that is where sort of faith comes in, and maybe science is a way of working in that regard. So I answered more than your question, and I didn't answer.
And you were looking for others, weren't you, in the chat as well? Okay, yeah. Jacques, how about you take over? I think you're muted there. You're muted there. Okay.
Yeah, thanks, Giles. I think we're getting pretty. It's now 2 hours into this session. I think maybe unless the panelists have questions and the lecturers have questions to each other, maybe one or two, we can engage that. But otherwise, I would suggest, Giles, that.
Sure. I mean, I just say that there's so much interesting this, and where can people find more about a career in science? Perhaps Virginia and Fathi, and then Chris can just say where to go, and then we'll have to wrap that up. But how do you do that?
I would just say, reach out to researchers, people like my own lab, and just say, hey, even if you just volunteer to help write a review paper with a lab, and say I wanted to review the literature regarding cardiology and spaceflight. I had a med student who just was interested in both, and he wrote a paper, so we wrote it together. Reach out to scientists that you like. If you see a paper you like, reach out to the authors and say, hey, I liked your paper. People always like to hear, oh, someone actually read my paper and they liked it. And say, you'd be surprised how rare that actually is. So just reach out to people. And if you like something, reach out to the person who published it and say, this is great. How can we do something together and just start to take initiative and start to do it, I'd say.
Sure. Virginia, very quickly.
So ISU has a number of professional development programs of various durations. You can check out any of those. And I would also encourage folks to check out all of the space workshops and conferences that are now virtual. It's easy to tie into a whole lot of different space events if you're interested in space.
Great. Fathi? And the Blue Marble, something about Blue Marble perhaps. What's going on there?
Yeah, Blue Marble is a non-profit organization, and indeed it's an organization that enables students from all around the world to participate to Mars missions, and so not just on human aspect, but also in origin of life, astrobiology, and so forth. So certainly I can look it up. Actually, I am an alumni from the ISU, so I did the program actually 20 years ago. I just organized the 20th anniversary of our program. And interestingly, one of our classmates is actually Jessica Mia, and she just flew to the International Space Station. I guess she came back last March, and so we were very proud of her mission. And as you were aware, I know she was part of the duet, which was the first time there was all-female EVA, and so that was very, very, very exciting. I think from a student perspective, I think as Chris Mason alluded into, reaching out to scientists that are working on specific topics of your interest is very important. Some space agencies do have programs for students, either as interns. For example, within NASA, we have programs for interns. Also, we have postdoctoral fellowships. And actually, what is amazing about it is that it is actually open to everybody. So it's not just for the citizen. It's anyone from around the world can apply to those fellowships. And so I will recommend to the audience, if they're interested in postdoctoral fellowships, to kind of look at those programs. And so again, within every nation, there are space agencies that are currently, looking at talented students. And I will remind everybody that the European Space Agency is not just opening for astronauts. And so I recommend all the Europeans that might be interested to apply. And so my understanding is application will start very soon.
Well, thank you very much. But don't mention the word Europeans. It sort of upsets me a little bit. I really would like to thank everyone. We're going to have a debrief meeting, and we're going to probably talk later on this, but I really would like to thank everyone for taking the time to participate in this session and this discussion. I'd particularly like to thank the I3 speakers. That's Chris Mason, Michael Waltemathe, and Kira Bacal, and of course our guest panelists, Fathi, Karouia, and Virginia Wotring. Thank you for the organizing committee of this initiative. That's Jacques Cohen, Peter Nage, Thomas Elliott, Scheista Sandrudin, Marianne Cervantes. There's a tremendous amount of work, obviously, that goes on behind the scenes. Thank you all for the huge amount of time that you put into this endeavor. There's a more complicated matter, which is, of course, collating all the questions. And we'd like to thank our triage team today, which was Dower Berger, Marianne Cervantes, and, of course, Anna McLaughlin.
Well, thank you, Commander Palmer. That was a great session that you put together. You didn't thank yourself. You really should, because you did a fantastic job putting this together. And the last one, and the sessions in between. Most of these sessions are your brainchildren. So really well done, and thank you very much. For those of you space cadets out there, don't forget to rate our lectures and our lecturers on our website. When you do this, you can win a prize. So do this, please. Our next session is next week, regular session time. It's Tuesday again next week at 3 p.m. Eastern Standard Time. It's noon in California, and that's 9, so of course 2100 Central European Time. Sorry for that, Giles. In the UK, I think that's 2000, right? So it's 2000. It's 8 p.m. Yeah. So the next session is called Personal Genetics, and the moderators are, and this is put together with the assistance of Helen O'Neill, Dr. Helen O'Neill, and Dr. Sissy Wamaita, and the first lecture is on CRISPR genome editing and how that is all done and why that is done by Helen O'Neill, and then 2 more lectures, one by Gregorio Alanis-Lobata, and that's about frequent loss of heterozygosity in CRISPR-Cas9 edited early human embryos. And then the last one is by Dr. John Appleby, CRISPR, IVG, ART, and OMG. Untangling the Ethics of Radical Innovations in Assisted Reproduction. And with that, Space Cadet, that's it for today. Live long and publish.